When skin sags, brain gets slacker. Losing the youthful firmness and elasticity of skin is one of the first outward signs of aging. It's not just our skin that starts to sag with age, but our brains too. New research from Newcastle University, UK, in collaboration with the Federal University of Rio de Janeiro, investigated the way the human brain folds and how this cortical folding changes with age. Linking the change in brain folding to the tension on the cerebral cortex, the team found that as we age, the tension on the cortex appears to decrease. This effect was more pronounced in individuals with Alzheimer's disease. Publishing their findings on October 24, 2016 in the academic journal PNAS, the team say this new research sheds light on the underlying mechanisms which affect brain folding and could be used in the future to help diagnose brain diseases. Dr. Yujian Wang, of Newcastle University, is the lead author. One of the key features of a mammalian brain is the grooves and folds all over the surface, a bit like a walnut. But until now no one has been able to measure this folding in a consistent way. By mapping the brain folding of over 1,000 people, researchers shown that our brain's fold is according to a simple universal law. The tension inside the cortex decreases with age. In Alzheimer's disease, this effect is observed at an earlier age and is more pronounced. The next step will be to see if there is a way to use the changes in folding as an early indicator of Alzheimer's disease. The expansion of the cerebral cortex is the most obvious feature of mammalian brain evolution and is generally accompanied by increasing degrees of folding of the cortical surface. In the average adult brain, cortex of one hemisphere was unfolded and flattened out. It would have a surface area of about 100,000 square meters. The folding of the cortex is regardless of size and shape, they all fold in the same way. We can use this same law to study changes in the human brain. From this, we can identify a parameter that decreases with age, which is interpreted as changing the tension on the cortical surface, like skin. As we age, the tension drops and the skin starts to slacken. It has long been known that the size and thickness of the cortex changes with age. The existence of a general law for folding shows us how to combine these quantities into a single measure of folding that can then be compared between genders, age groups and disease states. The team also found that male and female brains differ in size, surface area, and the degree of folding. Indeed, female brains tend to be slightly less folded than male brains of the same age. Despite this, male and female brains are shown to follow exactly the same law. This indicates that for the first time, we have a consistent way of quantifying cortical folding in humans. Throughout the lifespan of healthy individuals, cortical folding changes in the same way in both men and women. But in those with Alzheimer's disease, the change in the brain folding was significantly different. More work is needed in this area, but it does suggest that the effect Alzheimer's disease has on the folding of the brain is akin to premature aging of the cortex. Thank you.